All right, so let me put you back into the big picture. We're talking about passive transport, and there are two types: passive diffusion and facilitated diffusion. Osmosis is an example of facilitated diffusion. So in facilitated diffusion, solutes are moving down, solutes or water are moving down their gradient. That means moving from high to low. So that's from high solute to low solute in the case of diffusion, or from high water to low water in the case of osmosis. So the key part here is the facilitated part. Remember, that means helped. There are helper proteins that do this. So osmosis has aquaporins that let water flow through the membrane. Aquaporins are an example of a channel protein. So there are two types of proteins that help with diffusion, either channel proteins or transporter proteins, which also go by this word carrier. Your textbook uses transporter, but I really prefer the term carrier because I think that is a little bit easier. It gets away from having to use this word transport for everything. So we'll take a closer look at those two proteins. A channel creates a passageway directly through the membrane. So it really is like a tunnel. So we can see this example here. There's an opening. The protein just is actually embedded full through the membrane and creates this opening that the solute or water can pass through. This is an example of a gated channel protein. So that means it has an open and closed conformation. So this is about shape. So sometimes the protein is shaped like this, sometimes it's shaped like this. There's different triggers that can cause the gate to open. And this is a really fascinating subject that we don't really have time for, but this is a lot of what happens in your nervous system to transmit signals, is the opening and closing of a gate. So as I mentioned, aquaporins are an example of a channel protein. They sit in the membrane and are always open. So aquaporin, kind of drawing it in here, is just going to always let water pass through. It's not gated. And that's why, as I was saying, the cell is going to freely allow the movement of waters because that aquaporin is not gated. A carrier protein is a protein that's going to flip or change shape to carry. So this is why I like the word carrier protein better. It carries solutes across the membrane rather than being a channel. So, it's like ferrying it. It's also like a ferry. It picks it up on one side and delivers it to the other side. So here's the solute. It goes into the protein. The protein flips, so it changes shape, and then releases the solute to the other side. So I kind of think of it as a little bit, and I'll use my hand here if you can see it. So it takes the solute. My pen's going to be the solute. Solute comes in. The protein flips and releases the solute to the other side. So again, it's rolling downhill, hits the protein, causes the protein to change shape, and then the solute comes out the other side. And that's the way that carriers work. A lot of molecules are moved this way, especially the larger organic molecules like sugars, amino acids, and nucleotides that have to be moved in and out of the cell, but cannot directly pass through the phospholipid bilayer. So there's different types of transport proteins or carrier proteins. So again, remembering that while your textbook uses this transporter term, I also think the carrier term is helpful. And so you would see both presented on an exam. So the example we just looked at is an example of a uniporter. Uni is a root that means one. It moves one solute in one direction. So here's an example. The little purple dot comes in, flips, and comes out. So that's a uniporter. A symporter moves two or more solutes in the same direction. So sim means same. So the red dot comes in, the blue triangle comes in, and they both go out the other side. So they both got moved from top to bottom. That's the same direction, even though it was two or more solutes. An antiporter, anti means opposite. So two or more get moved in opposite directions. And so in this case, the blue dot gets moved down, whereas this little, I don't know, maroon color shape gets moved up. So the same protein picks up one thing and moves it to this side, and then picks up the other thing and moves it to the other side. So that's an antiporter. Two solutes move in two different directions. So I'm going to just refresh your memory here again about passive and active transport. These are all examples of passive transport. We either have passive diffusion, where the solute passes directly through the phospholipid bilayer, or we have facilitated diffusion, where the solute has to either pass through a channel protein or a carrier protein, always moving from areas of high concentration to low concentration. This is called going down the gradient. It's analogous to rolling downhill because it does not require any energy. In the next video, we'll talk about active transport, where solutes are moved against their gradient from low to high concentration. This is analogous to going uphill and will require an input of energy. And in this diagram, you can see that that input of energy is coming from ATP, which is an energy source in the cell that we're going to begin talking a lot about in this chapter and in future chapters.